concept that they're very selecting the data molecules from the one of the
Thank you. Nice to meet you. Thank you for coming.
Hi, how are you doing?
Thank you for hosting.
Oh, sure.
Yeah.
Sure.
Thank you.
So you all set up?
Yeah.
You got the microphone?
Yes.
But
like a 2 to 3 projects. Yeah, I think I schedule somewhere, but okay.
Already been and circularity. Yeah, so Alzheimer's. Oh yeah, yeah. We're doing like RNA. It's Monday, Wednesday, so it's tomorrow.
Yes. The responsive here.
After dinner, I have the rehearsal, and
he's ahead of a teacher.
I can't remember the rehearsal.
He was quite upset. Yeah. He was so distracted. Yeah, I wrote to him. Yeah, like, how are you doing? We were going to talk about the ES3L project. He had this reason out on this causing sterility phenotypes of priority levels. They talked about the project never got into. How was that?
Okay.
Title 6. That's why I was so glad to come up.
Yes, thank you. So it's been a challenge to show them.
Well, there's the back for that. That's a cheap. That one has very clear versus. It's quite crowded in there. Yeah.
Yeah. Oh, that's so cool. I'm not presenting.
Who knows though?
That's so cool.
See this live
Like in July.
Okay, that's pretty
So
cool.
I think she's,
Is she excited?
yes.
Yeah, of course. That's obvious.
All right, well, I guess we can get started. Thank you so much for everybody for coming. It's an absolute pleasure to introduce Vicky Yao for this Genome Science Institute seminar. Not just because we have been very close collaborators and friends for more than 10 years, but also because her research is incredibly innovative and exciting. So even if Vicky presents as a computational scientist, it didn't start like that. And her first experience in biology was in high school as a high school research intern at NIH, when she was doing bench work, but actually not like what you could expect, mini preps and such. She was actually identifying topologically associated chromatin domain with 3C in PhD Zhao's lab. And so effectively having a cooler research portfolio at the age of 16 than I had as a postdoc when we first met. Pretty cool, but she still decided to terminate her career as an experimentalist at that point. She went to U Chicago for her undergrad, majoring in economics, minoring in computer science, obtaining at the same time a master's in statistics. And she had found her specialty, which led her to Princeton, where she obtained a PhD in computer science under the supervision of Olga Troyanskaya. And this is where I met her when I was at Rockefeller in the lab of Paul Greengard, and our two PIs had the great idea to match experimentalists and computational scientists from their respective labs, and so we kicked off a collaboration that is ongoing ever since. Vicky just impressed everybody in Paul's lab with her creativity, her deep knowledge of neuroscience, and her talents conveying complicated functional genomics concepts to biochemists. She really thrives in collaborations, and she was equally comfortable establishing exciting analysis framework for our mouse selective neuronal vulnerability data to identify cell type associated with rheumatoid arthritis flares in humans with Bob Darnell, also at Rockefeller, or for using worms to identify gene networks associated with longevity with Colin Murphy at Princeton. Without doing a postdoc, she directly started her lab at Rice University in the computer science department, where she designs new analysis frameworks to extract biological meaning from big data in neuroscience and cancer. She still always works closely with experimentalists, keeping disease relevance as her compass, and always strives to provide actionable insights in biology, identifying genes and pathways that can constitute the basis for follow-up experimental work. She became a scholar for the Cancer Prevention and Research Institute of Texas in 2019. She obtained an NSF Career Award in 2022. She's involved in many outreach activities, in particular with groups promoting data science among female students. And she's an accomplished educator, teaching undergrad and grad classes in computer science, both bioinformatics or more broadly. And without further ado, let's welcome Vicky, who's going to talk about integrative methods for the analysis of complex traits and diseases.
Thank you, JP. I think I haven't had people mention my high school foray into experiment for a long time. But yeah, so today I'm going to talk to you about, we'll see how many projects we get through, 2 to 3 projects that we've been working on. So the first one I want to talk about is integrating population scale disease signal with high resolution single cell transcriptomics, hopefully to enable new biological insights. And then the second project that I wanted to talk about today is uncovering how alternative splicing may rewire protein interactions to reshape disease pathways, and look at how we're applying this to analyze TCGA cancer data. And then lastly, if we have time, there's like a small kind of more computational project that we've undertaken to improve gene set comparisons, essentially taking advantage of some of these really incredible machine learning advances to basically, help improve one of the most fundamental bioinformatic tasks we do all the time, gene set enrichment. So if we have time, I'll quickly talk about that. So this first project, thinking about population scale disease signal and single cell transcriptomics, was really motivated by, some of you probably are already familiar with these giant GWAS studies that have been enabled since Human Genome Project was completed, where, great idea, right? So it's like now we can look at different variants between people. If we have different things that we're interested in, a disease, a complex trait, grab a bunch of people that have such, a trait, and then compare them with controls, look at different variant frequencies. We could do this for Alzheimer's disease or Parkinson's disease, depression, schizophrenia, college education. Again, it doesn't have to be a disease. And then the idea is now we're going to be able to really better understand risk factors or the genetic basis of any of these complex traits. Of course, as we know, there have been a lot of important findings driven by this GWAS data, but at the same time, there are still multiple gaps, right? I think a lot of the genetic bases, we found a portion of it. There's it seems like some question of this inheritability where there's a gap between what we've been finding in GWAS data. But another interesting thing that have intrigued us is a lot of the diseases or even complex traits, we can think of them as kind of tissue or cell type specific, right? We know a lot of the examples they gave are actually neurological. Then there are complex traits like college education where we're like, there might be some tissue or cell type specificity, but it's vague. Like, what should it be? Meanwhile, GWAS data, really, this is like looking at genetic variants, right? Like, most of the time germline variation where there's no information regarding tissue or cell type specificity. So we have this sense of vague genetic risk or genetic kind of predisposition for some complex traits. Yet we're we might also be very interested in the cell type tissue specificity of such traits or diseases, and there's an interesting gap there. And meanwhile, I think I focus a lot on the neurological disorders here because actually GP was one of the first people that helped kind of convey to me the kind of incredible complexity of the brain. So of course, we know that there are lots of brain regions. Brain regions do very, very many different things. And then within brain regions, we have lots of different cell types. But then at the same time, these cell types are actually spread around these different brain regions. So, I'm not the one to probably talk about this with all of you. A lot of you are kind of much more experts in terms of this. And we know that, we can think of them as often in these different broad classes, support cells, neurons. They're spread across these brain regions, and yes, we know there's region specificity in disease, we know there's cell type specificity in disease. How do we kind of reconcile all of these different aspects? And so there could be an interesting opportunity here, especially as people started to do all these giant single cell transcriptomic analyses, right? So it's like now, in say from the normal brain, people are doing this systematically in mouse, in human to some degree, where we can collect lots of single cell expression that gives us this very high resolution profile of all of these neurons or support cells, glial cells, as well as across brain regions. So we were motivated by kind of a simple question. So we have this non-cell type specific giant population scale data. Meanwhile, in kind of a much fewer number of individuals, we have some sort of notion of the normal-ish expression profiles of incredibly high resolution cell types. Can we integrate what we know about the disease variation at population scale with these high resolution, but of course unpaired expression profiles? This unpaired can go very far, right? We know that the brain data, at least in the early days, was mostly collected on mouse, and it's much higher quality in general than the human data. So when I say unpaired, I'm saying, we have, population scale human disease data. We could look at a couple of humans that weren't as part of that collection at all. But we might also be interested in actually comparing this kind of large scale population scale data and look at, can we actually use the mouse brain and actually kind of derive some insights into what we're seeing, at population scale in human. So we were not the first ones to think of this. There's actually been other work looking at this idea where, we can take GWAS data. So I'm not going to go over the details of this process, but just kind of one note here is that GWAS data is looking at, genetic variants, single nucleotide polymorphisms in the genome, right? So these are variants. They might be spread out kind of all across our genomes, but we need to, for downstream analysis comparing, say, with single cell data, we want to collapse it to some kind of a gene unit, right? So there are other tools that have been developed for this. So one such tool is Magma. And so I won't talk about the details there, but it's kind of a high-level tool that considers, the LD kind of linkage disequilibrium between variants, collapses what we see about disease signal at the gene level, and then people kind of combine these data types in different ways, but we combine Magma with single-cell RNA sequencing, which gives us cell type specificity, and then people try to assess some kind of cell type trait association. When we kind of looked into the current methods, we found that as a computational person, there were a lot of things you can tweak and tune about these things, and it was kind of brittle. So for one thing, most methods require some kind of thresholding at this magma z-score, and we'll get back to why thresholding is really challenging, because you can imagine, we can think of different diseases that might involve different numbers of genes. We don't actually know what that should be. We can kind of tweak that. People also try to do different thresholding at the single cell RNA sequencing level, which again, we already know that probably for different cell types, you might have different thresholds there for different types of expression. The decision space actually explodes quite a lot here. And then there's like kind of some methods that to assess statistical significance make heavy use of simulations. This is much more of a computational point, because what this means in practice is as you have very large single cell data sets, it just is computationally intractable. Basically, you actually can kind of try to assess, if you think about making an analysis with every single cell type in the brain compared with a particular trait, the computational kind of tractability kind of drops very quickly. So, actually I think JP was one of the inspirations for this project, because he was like, we have some interesting observations, which I'll get to at the end, in terms of Alzheimer's disease, GWAS, and cell type specificity. Is there something better we can do beyond kind of the current state-of-the-art tools? So Qiliang Lai is a graduate student in my group that has been working on this, and this is in collaboration, of course, with JP, where we were like, can we come up with something that fills in that box for how we can integrate single cell data with GWAS data in maybe like a different way that better outperforms current state-of-the-art tools. So I'll go over this kind of quickly, because then we'll dive into kind of some of the details of the method. And the idea here is, one thing that actually has been a gap in a lot of other tools is a lot of tools will look at just trait and cell type association, ignoring brain region. And as we know, there are a lot of brain disorders where brain region actually matters a lot. So we wanted a method that allows us to more flexibly select different levels of what we're calling granularity, which includes either kind of high resolution characterizations of the cell type, or combining, say, cell type and brain region information. So this is just the idea that instead of just taking out of the box, like kind of the immediate high level annotation of a cell type, we can actually look at something kind of much more high resolution. So we develop a computational method to calculate what we are calling a specificity score that we believe better characterizes the kind of gene and cell type markers in a way, but actually has a better quantitative representation of a cell type. And we find that this is particularly important when we try to increase the resolution of the cell types that we're looking at. And then we use a statistical-based regression association method to kind of assign relevance between the disease that we're looking at, or the trait we're looking at, with single cell data, look at statistical significance, and then we actually developed a completely new framework that other methods have never kind of deployed on such a framework, in terms of, a lot of methods end with, we think there's an association between this cell type and this trait, and which is valuable, but we have the next level question is, if we find an association between a cell type and a trait, can we go one step further and be like, what are the genes specifically that have driven that association that we've discovered? And of course, we see that as like a hypothesis generation machine there, where we can actually then not only understand whether a cell type and a trait may be associated based on all of this data, but which genes, or ergo which pathways, are driving that type of disease risk. So as a computational person, we often slap on some equations to talk about the specificity score. The details aren't actually critically important. What I'm trying to show here is that we actually consider two aspects of the specificity score. The first aspect is actually almost like a z-test for expression quantitative differences. So what there's two components of our specificity score. The first component is there there expression of the gene that we're looking at, right? But in single cell data, we realize there's actually a secondary component. What is just is the gene there? So we actually wanted to develop a specificity score that captures both of these things, the binary presence absence of the gene, as well as the gene's quantitative kind of expression, taking into account the level of expression. So we come up with this kind of new score. It's kind of in the end actually quite simple, that combines these two things. And we'll talk about why, I'm happy to answer or discuss intuitively why we think that combining these two things together is better than, say, considering only differential expression or binary presence absence of the gene. So before we move on to real data, what we really wanted to do first, and what a lot of people who develop computational methods do first, is how can we evaluate this method when we don't actually have a ground truth, right? If we think about disease, cell type associations, we're trying to do discovery, but we don't know. The point is, like, a lot of these diseases we don't know. We can check if it makes sense, but what we can do here is actually just do lots of simulations. So first thing that we can do is say, if we've destroyed all biological signal in a data set, how often will we call things significant, right? So this is saying, let's scramble, say, the z-scores or the expression data. We would hope that we come up with a statistical significance estimate of 0.05. That would mean that you don't actually get false positives more often than that. So in computational terms, we're saying that we want this method to be well calibrated, and so what the p-value we assess is actually reflective of the number of false hypotheses that it proposes. So we see, seismic is our method at the top left here, and the other three methods are basically what's the current state of the art, so to speak, in terms of doing this type of cell type trait analysis. And so the way to read this plot is on the x-axis, this is kind of the expected kind of number based of false hypotheses based on kind of the p-value that you're reporting. On the y-axis is what you're actually observing. We are hoping that we lie along this diagonal line. And you might be like, why don't we want to be so FUMA, for example, if you look there, has the line much below the dashed line. In that case, it's actually reporting, it's what we're calling conservative, right? So when it says it's reporting 5% false hypotheses, it's actually reporting fewer than that. You might be like, that sounds actually like a great thing to have, right? You actually are even more conservative. The difficulty here is that there's always actually a trade-off between how many false hypotheses you kind of are reporting and, of course, the power of your method. And so what we see and what we would like to see is seismic that's along this dashed line, which means when we say around 5% false hypotheses, that's around kind of how many that you're actually seeing. And the trade-off here really kind of is more relevant when we look at type 2 error assessment, which is just the power of our method, right? So in our simulations, we can artificially perturb certain genes at different expression log fold changes. So we're just saying, of course, once you have very high kind of expression changes, lots of methods start being able to detect that better. We actually see that two of the older methods, FUMA and MAGMA, have a lot of trouble when we kind of deploy a more complex simulated framework. This other method, SEDRS, that we see is a little bit inflated in the type 1 error assessment, if you see, but it's actually, the average is right on that line as well. Also has more power, but especially seismic, has a lot of power at the lower expression level changes. So this is all simulated data, right? We're not talking about real data yet, but we also wanted to look at the computationally efficient angle. So SEDRS is the method that I mentioned that uses a lot of simulations to assess statistical significance. So this is not as critically important in the moment, but the idea is just that, as single cell atlases grow very large, it is, take an incredibly long time to get results. Whereas seismic is comparable to some of these early methods. So before we dive back into the brain example, what we actually wanted to do is take kind of a snapshot of single cell expression across the whole body. And so this is from Papillomiris, which again, if you're not familiar, is the single cell atlas that people have kind of basically systematically dissected a mouse up into all of it, the big organ systems, and done tons of single cell transcriptomics. And we can actually compare this with 27 different GWAS studies that we've kind of obtained. And so the way to look at this is on the x-axis here are all the different cell types. We've actually then siphoned them also by kind of organ system. So if you were to be able to look kind of more closely, you'll see that there are certain cells that might kind of be present in multiple organ systems. We've separated them. And then on the kind of y-axis here are rough categorization, essentially, of these GWAS studies that we've grabbed. So we have, a couple of neurological disorders, immune-related ones, and then a very broad characterization of just other, where it wasn't, quite clear, say for smoking or BMI, there's not like some totally immediately coherent characterization. So I think just looking at this, we saw it's kind of nice to see, roughly, what we might expect. So a lot of the blood immune kind of cell types were, called as kind of really enriched or associated with immune-related disorders. And then for neurological ones, we see a lot of brain associations. Interestingly, we also see some pancreas relationships there, which I think has been supported somewhat by kind of metabolism or some other processes kind of relating to neurological disorders. We also see kind of more subtle things like glucose or HDL, LDL levels associated with liver. So I think this was mostly like I think there are some interesting associations here that we could already follow up on, but it was more like do we think that the method is making reasonable biological sense? Because what we actually, set out to do is actually try to look deeper into neurological disorders and look for some type of, interesting new biological insights, right? So before we kind of jump in further, I wanted to kind of look at this Parkinson's disease example where we grabbed Parkinson's disease GWAS. And so we did a case study here because, Parkinson's disease has like a very well-known association in terms of dopaminergic neurons. And these are the neurons that are very vulnerable in Parkinson's disease. But within kind of the brain, we know that especially kind of dopaminergic neurons in the substantia nigra versus dopaminergic neurons in other brain regions have particularly high kind of vulnerability to the disease. So I mentioned that traditionally a lot of methods choose a very coarse resolution when they're doing the analysis. So what we're showing is something kind of reminiscent of a lot of these types of GWAS trait associations kind of studies. We'll take like kind of just look at them at the kind of neurotransmitter level, for example, and say, do we see dopaminergic neuron association with the trait? And we'll see that actually several methods, so of the newer ones, HUMA, SCDRS, as well as seismic, all find a significant dopaminergic neuron association with the disease. And so this is expected. This makes us kind of, we see that seismic is, particularly powerful in doing this, but this is kind of to be expected. But I mentioned, right, that we were actually interested in, can we do something more interesting in terms of look at kind of higher and higher levels of resolution to kind of derive kind of deeper insights, right? So the dopaminergic neuron association with Parkinson's is well known. So what we actually then did is actually look at not just, these are the categorizations are roughly kind of just the original terminology based on kind of the annotations are derived based on the folks that did this mouse single cell transcriptomic data set. And so that's subclass, brain region plus class, brain region plus subclass, and then brain region plus cluster, as well as a much finer resolution brain region plus fine cluster. So we'll see the numbers at each level actually indicate how many categorizations, or think of them as cell types or cell type characterizations there are at each level. So if we're talking about looking at kind of this very, very coarse subclass level, there's actually only 14 different cell types we're comparing. So yeah, dopaminergic plus say GABAergic and then like microglia or some other cells. Then we can go down to brain region plus class, where class is actually lower resolution than subclass, right? So we're just saying like neurons, like it's like a big, where all the neurons have now been collapsed together. And then, so you can keep going kind of with more and more higher resolution until this last one where the everything is the most specific is where we're looking at brain region plus fine cluster. So what we see is that seismic actually is the one that can work really well in terms of increasing the level of specificity and actually still finding the dopaminergic neuron association, but at clearer and clearer kind of, parsing out kind of interesting subgroups. So the rationale for this, which I didn't talk about what the other methods are doing, but the difference is a lot of the other methods are looking at just mean expression values or just the raw expression values. So this is where I want to highlight that when we calculate this kind of specificity score, you can imagine that the problem with not calculating a specificity score is when you increase resolution, a lot of these neuron types are actually quite similar, right? You can imagine that maybe there's like a nudge, a slight differences here. So when you have increased the resolution to 231 different cell type characterizations, at the mean expression kind of representation, they look quite similar. And in fact, then they're competing kind of, once you do FDR correction, is really when you kind of just completely lose a lot of signal. Meanwhile, because seismic is calculating a specificity-based score, we can actually look for subtle expression differences that are consistent across, whatever cell type characterization you've chosen, and that helps kind of us maintain more power or distinguishing ability across cell types, even as we increase the level of specificity. So we actually find then fine-grained kind of associations between the substantia nigra neurons that basically purportedly make sense in terms of associations with Parkinson's disease. So moving forward from Parkinson's disease, we're also really interested in actually examining Alzheimer's disease associations using seismic. So a note here, so in a lot of Alzheimer's disease GWAS, so what people have been doing is actually grabbing a huge amount of Alzheimer's disease, clinical Alzheimer's disease patients. Clinical meaning they've been diagnosed based on clinical presentation, and basically what people have been consistently finding is when you do these Alzheimer's disease GWAS, you find genes associated with microglia. And so people actually have increased the size of these cohorts for these population scale studies larger and larger to maybe 1 million patients, and yet the microglia signal is the strongest one that becomes clearer and clearer. So I want to mention this because, of course, microglia is the support cell, and yet it almost seems like kind of a paradox because in Alzheimer's disease, we know, yes, there's an association with microglia, but a lot of JP's work actually, and many others have shown us that there's incredible kind of regional and neuronal specificity in Alzheimer's disease. So early on, even pathologically, people found there are particular neurons in a particular brain region that die much early on. So the entorhinal cortex neurons are particularly vulnerable, yet the GWAS genetic kind of basis keeps showing us a glial cell, and we'll see that this glial cell, the microglia, there's not much regional specificity. Maybe, yes, hippocampus, but has the strongest association, but really there's actually not that level of very specific regional association that we would expect to see. So the question we had then was now that we have our seismic tool, could we better kind of, find maybe, look at, several different Alzheimer's disease GWASs and figure out, is there something, what might be driving this? And so what was actually kind of exciting for us is when we looked for, used a much, much, much smaller GWAS study, but instead of, diagnosing patients or assigning patients to the Alzheimer's disease group based on clinical presentation, the researchers in this study used actually cerebrospinal fluid tau levels, so a pathology based or a biomarker based, like a quantitative kind of trait, applying seismic to the same exact expression data. We actually do find neuronal signal, and in fact, several of the top kind of associated cell types were exactly the neuron types that we know are vulnerable in Alzheimer's disease. So we actually thought this was actually quite interesting as like a meta insight that perhaps sometimes bigger is not always better, right? If you take a giant GWAS study that is grabbing a lot of Alzheimer's disease patients that have just been diagnosed based on memory or behavior, perhaps there's like a lot of heterogeneity there that might be driving that. What's common between all of these patients is some kind of a microglia association to the disease. Meanwhile, if we look at, this is like many orders of magnitude smaller GWAS studies, so these are like thousands of patients versus tens or hundreds of thousands of patients, right? We actually can find much more specific signals. And so, as I mentioned kind of early on when I was discussing the seismic method, if we look under the hood, what we're actually really doing is kind of a regression-based analysis comparing the specificity score to the gene risk. And what we are doing is actually basically looking at, is the slope, or is the line that you draw more or less significant than you'd expect by random. What this enables us to do is actually each of these dots is actually a gene. So we can actually use kind of what people have developed in statistics in terms of looking for what's called influential observations. So which genes are the ones that have outsized influence on the significant result that we find downstream. So that's actually why it's called DF betas, where beta you can think is that betas are common, kind of the Greek symbol we use for the coefficient for that regression. So when is beta really different? Approximately we can think of it like that. So in a more kind of tangible example for the CSF tau GWAS, for the significant association we find between the medial parietal cortex neurons and the GWAS, we can basically essentially calculate what this DF beta's influential score is for every single gene. And so I've highlighted the dark red are actually genes that are common between two different kind of entorhinal cortex regions, their association with CSF tau GWAS. And of course, then we've been in discussion with JP to further analyze or interpret some of these results that we're finding in terms of these are the genes that seem to have outsized influence on potentially influencing the risk between these vulnerable neurons and GWAS data. So just to wrap up this project very quickly, in terms of the seismic method that we've developed, just want to highlight that what we're doing here is integrating single-cell RNA sequencing data with GWAS to hopefully pinpoint or highlight disease-relevant cell types. We use under the hood, a novel cell type specificity scoring method to link these genetic risk factors to cellular phenotypes. And what we're really excited about is this new way of thinking about these types of analyses, what we're calling influential gene analysis, that can inform hypotheses for genes or pathways that drive the different disease cell associations. Sorry, before I move on, does anybody have any questions about that first portion? Yes.
So have you only used seismic to link single cell data to a physical trait, or have you used it for other purposes as well?
So you can use it to link with any GWAS study, right? So basically, if somebody's done a GWAS on it, we could apply seismic to it, as long as we just basically choose the GWAS study and the single cell data. So that's why then, that's why I was like, college education, like a complex trait that people have studied using GWAS. Yes?
Did the other tools also link all these common GWAS traits for the brain and the pancreas?
Yeah. So actually, several of them do. Actually, sometimes at weaker kind of p-values. In the paper, we have it in the supplement where we have the same heat map basically over and over again with all of these different methods. What we find is that a lot of the associations we find actually are overlapping, but we basically find kind of we kind of boost up some of the power of certain traits, and then we do a kind of a more deeper dive analysis on all the combinations of the results that we're finding there. Okay, cool. So the next method I wanted to talk about, so we're going to change gears a little bit. So I've been really interested in thinking about looking at kind of networks of relationships, and so we'll talk a little bit about how we're thinking about the downstream functional impacts of alternative splicing. So this is all motivated by one of the observations that so many of you might be familiar with this incredible resource that has been built up over time. PPI networks, or protein physical interaction databases, where people have systematically checked usually in yeast whether or not two proteins physically interact, right? So BioGrid, and there are a lot of other databases that capture what people have been studying, often through high throughput screens. But if you do grab a BioGrid PPI representation, it's a static kind of set of proteins that purportedly can interact in an yeast cell. So this is like, if we just grab that network and use it for downstream things, that network is always static, right? It's just like all of the possible interactions. So that's why we were like, PPI databases capture this static snapshot of what we call all potential physical binding interactions. Of course, there are many, many that are missing, depending on if people have screened them or not. But we also know that biologically PPIs are highly dynamic, right, in different cell types and different tissues. Again, we expect there to be different protein-protein interactions. So people have looked at basically overlaying, say, tissue-specific expression just on a PPI network to be like, oh, okay, likely we'll see a subset of what we're seeing maybe in a PPI database based on whether or not a gene is even expressed. So we were like, okay, is there anything else we can do in terms of combining the static prior knowledge with experimental data to capture dynamic PPIs beyond just saying subsetting the PPIs by which genes we see are expressed? So as a computational person, we have to first think, what data do we have to work with? So we have the prior knowledge. Number one, we have BioGrid and all of these databases that gives us a knowledge base of possible protein pairs that physically interact. So that, of course, very valuable information. But prior knowledge too, we actually know that a lot of protein interactions are frequently mediated by domain-domain interactions, right? So we know that people have put in a lot of work to call protein domains within proteins, and these protein domains are important functional units that actually mediate a lot of interactions. And then number three, there are actually a lot of, there's also databases that characterize, that tabulate previously characterized domain-domain interactions. So, thinking about these three elements together, we kind of we realized that splicing is actually one of the main mechanisms that can mediate protein domain inclusion or exclusion. So now the next idea was, okay, can we then systematically kind of keep track of all of these pieces of information for any new experiment that comes, we can look at maybe what splicing changes are happening, and ergo what protein domains might be changing, and then which protein-protein interactions might be changing. So this work was led by Ruth Dannenfelser, data scientist in my lab, and so we call this method split-P. So this is a common thing you'll see with computational methods. We try to wrangle some words into having some kind of an acronym that roughly matches those letters. So this one was a little bit harder. So you see that's why we have some random letters in words that are capitalized. So first, we grab all of the databases we can to build a large PPI network that represents all we currently know about which protein interactions are happening. We can again now keep track of, we can call all of the protein domains in any pair of proteins and be like, okay, these are the DDIs that have been kind of reported before. So we can say that the relationship between protein 1 and protein 2 is likely mediated by, we don't know which one or all of them, these domain-domain interactions within the two proteins. So now if somebody brings us an experiment, we can look at, oh, okay, if we know that a particular exon increases or is more often found in the isoforms present in that data, then perhaps that domain is kind of more present. And so we would be calling that as, maybe the interaction, it's possible that the protein interaction is, say, stronger, or if maybe that exon didn't exist before, the protein interaction was much weaker. So we've now kind of potentially gained an interaction, or that interaction has become more fortified. Meanwhile, we also can see in a case where an exon decreases, say domain 3 in that protein 1, and we know that is kind of supporting two domain-domain interactions. Maybe it might, so loss and gain are kind of stronger words, but that protein interaction that maybe previously existed is now weakened, or perhaps even destroyed. And of course, that means we have the third case where we may have some exons that are increasing, some exons that are decreasing. We don't know what that ends up doing necessarily, and we call that just chaos. We don't have a good prediction on what the final outcome of that, do they cancel out, or what happens downstream. So in this case, we wanted to first kind of examine the method in the context of cancer. So people have observed this in the past, but if you look at, so we were able to grab a lot of TCGA data. This is nearly 8,000 tumor samples across a bunch of different cancer types. So we actually hijacked the commonly used kind of plot for somatic mutations. This is just looking at splicing events. So this is the number of alternative splicing events, actually more specifically when an exon is skipped within the different isoforms that are called for these tumor samples. And we see already that inherently different cancer types have actually different amounts of splicing, although splicing is occurring broadly across all of these cancers. And specifically, we see that these three cancers on the right here, esophageal cancer, ovarian, and stomach cancer, seem to have much higher numbers of events than other cancer types. The next thing we actually wanted to do, though, is we think a lot about tissue specificity in her group, and we already know also that different tissues actually have probably different baseline kind of amounts or different backgrounds in terms of how much splicing occurs. So next, we actually were able to basically grab GTEx data. So these, it's another large consortium, of course, that gives us nearly 6,000 normal tissue samples, and match them with the corresponding tumor tissue, and correct for the basically naturally occurring amount of splicing in the tumor sample. So if we look at what's actually largely differential between the tissue background and the tumor sample, we see that there's a slight re-ranking. And so what's interesting here is that the events, first of all, across the cancer types drop around tenfold. So we're saying that there are a lot of genes that are just generally spliced all the time. It might not be involved in cancer. So that's, if you look at the numbers on the y-axis, there is of course a big drop. That is to be expected. But we now see that some of these highest amounts of splicing cancers that we saw earlier, they're actually now not necessarily the ones that have the most splicing changes. We actually see, interestingly, several brain-related cancer types. So glioblastoma, glioma, these are now actually by far the two cancer types that have the most kind of number of splicing or skipped exon changes above the brain background. So that was, of course, the first immediate, most obvious observation, but we were interested in diving into this data a little bit more. So just to recap, what we're now going to do is we actually take, so this was an effort co-led by Jeffrey and Ruth in my lab. So across these 28 cancers, we have matched GTEx backgrounds to the TCGA cancer samples. We're now going to just combine it with the human PPI network and look for kind of these rewired gains, losses, or chaos edges, so that for each patient, we actually will generate a network of potential changes. So this means that, given our 8,000 or so tumor samples, we're now going to make basically 8,000 networks so that we can kind of look into what's happening downstream. So first, we actually looked for just simply how many rewired gains and losses do we see across these cancer types. One pretty striking observation that immediately jumped out is not only were the two brain cancers the ones that had many more splicing types, it was distinctly unique in that there were actually many more gains of interactions than basically all of the other cancer types. So even ones that had kind of similar kind of levels of splicing changes, the two brain cancers had many cases where we were predicting a potential gain of interaction between proteins versus losses. So so that's what I'm highlighting here. So we also then were able to look at which genes are the ones that are frequently being observed as involved in some kind of a rewiring, and we see that there's a core set of rewired genes that are conserved across cancers. These are these top 5 here. Genes that are bolded along the y-axis are actually known cancer driver genes, and so there are actually 3 very kind of popular or famous known driver genes in the top 5 genes, and then the other 2, TPM1 and TPM2, are actually also well documented for their role in splicing and isoform generation. And then the bar on this right is actually showing kind of what types of edges across all of these cancer tumor samples that they're mediating, and we see that a lot of these top rewired genes are all seemingly to be involved in loss of interaction, whereas then we basically are kind of diving into this data set, and it's very interesting to see basically which genes mediate these interactions and when is it more cancer specific versus conserved across a lot of different cancers. And we're hoping that this can help us kind of gain some deeper insights into how splicing might be affecting different cancer types. So I'm going to skip over some of the kind of nitty-gritty details of like some of these kind of meta-analysis that we're doing, and just mention that one of the things that we were very interested in looking at is how does this kind of translate to kind of survival or prognosis of the cancer patients. So we first looked at basically, this is a graph showing basically the number of splicing events. So it's that first graph where we showed all the squiggly lines. The y-axis now is on the x-axis. And then the number of edges based on basically our kind of patient-specific networks that we're generating. Because just because there are splicing events doesn't always mean that it translates into some kind of a predicted network disruption or network rewiring. So we were able to use a machine learning method to basically call outliers. So being outliers, there's actually relatively few per cancer type. But what we actually found that was quite interesting is that if, in specifically gains of interactions, we actually found significantly worse survival. This is both using the Kaplan-Meier analysis as well as a Cox hazards analysis that's been corrected for tumor stage, age, gender, and tumor type, where we actually still see that basically these outliers in terms of basically patients that have many more splicing events than you'd expect, they have significantly worse prognosis. We have the same analysis using loss of interactions, and that is actually not as significant. And so this is actually across cancer types. So there's something special about these splicing events that mediate gains of interactions that seem to be particularly bad for the patient. And we're also diving kind of deeper into this analysis. So some of the takeaways here is that we think that's, well, I think we're hopefully showing that splicing can potentially impact PPIs by affecting important protein domains that mediate the interaction. And cancer and tissue types can have vastly different splicing patterns, which may translate to a myriad of different functional implications. And hopefully what we're hoping to do next is kind of tabulate all of the results that we've seen at the patient level and enable to better inform our understanding of different cancers. And I wanted to save some time for questions, but I'll quickly mention that our work for improving gene set comparisons uses like a clever algorithmic idea, and the idea is that traditional gene set enrichment looks for gene overlap, and we're probably all familiar. You can do this using an overrepresentation framework, where basically you look at if we take random genes, how often do we expect them to overlap? Or you can do this in a rank-based way, right? Say you have differential expression log fold change. You're like, oh, okay, are they ranked higher or lower than you might expect? So our big insight here is that if you say you have an annotated Go term, and there's only, so this is a toy example, you only have X and Y, two genes that are overlapping between gene set 1 and the annotated Go term. What if I told you that A, B, C, and X, Y were actually kind of having lots of protein interactions, co-annotated to a lot of other things? What we might expect is maybe A, B, and C should really also have been annotated to that Go term, but just haven't been because people haven't studied it. So the big insight here is if we can use kind of what we're finding in terms of machine learning methods and looking at kind of gene-gene similarities in a functional space, can we actually boost traditional gene set enrichment analysis by leveraging, say, PPI information to find enrichment in things that maybe are poorly annotated? So that's the general idea. We have some background. I'll skip over this and what people typically do in this space. We have a motivating example here to say that what people have typically done is actually, if you want to use PPI representations, people often take the centroid or the mean of all of the genes as like a representation. And what we claim is that the centroid, especially for diverse heterogeneous gene sets, is a very poor representation because it loses a ton of information. So we just took the KEGG Alzheimer's disease genes, slapped them on top of this embedding representation of a PPI network. And we all know that a disease pathway can often have many, many sub functions. And so what we're saying is that if you actually look at many different Go terms, they're actually kind of spread all across this plot. And something like oxidative phosphorylation, there's actually a tight cluster of genes, but that's very far possibly from the centroid. So we developed the method to perform gene set comparisons in these embedding spaces. So this is actually just a machine learning representation of, say, the protein-protein interaction network while considering heterogeneity. And so this was led by Letruan and my group. So I won't go over the basic main idea is that instead of doing this collapsing to a centroid very early on, what we do is actually look for each gene in one gene set, we look for the best matching gene in the other gene set, and we can calculate a score for that similarity. And we can do this over and over again for every gene, in both directions. And then we do some kind of mathematical magic to kind of permutations-based way to calculate a statistical significance, and we can actually compare this with a bunch of methods. So yeah, I'll skip through this. And what I want to just say is that this method, which we call Andes, significantly outperforms basically any kind of baseline approach in identifying the relevant gene set. We can actually deploy this to traditional gene set enrichment, look for, so people have annotated in certain disease data sets what they expect in terms of which pathways should be enriched. And we can see that using the Andes approach, we can get much better basically associations or enrichments of the expected pathways. We also have extended it to the GSEA approach. So a quick recap here is that what GSEA does is, of course, now you have a ranked list, and what you do is actually computationally go down the list. Every time you see a gene, you kind of increase a score by 1. If you don't see a matching gene, you decrease it by 1. So then, when you kind of have a big mountain peak, so to speak, like with GSEA, you're like, okay, there was enrichment at some point. So we use a similar intuition, but instead of kind of incrementing or decrementing by like a 1 or a fraction, a weighted fraction, we can actually use, say, like a score based on how similar 2 genes are in PPI or functional space. And we use that same best match approach and basically hijack the GSEA method to do this. And what this also gives us then is we can actually, for maybe genes that are highly similar, none of them could be annotated to a Go term, and yet we can actually assess whether or not, say, the results of a differential expression analysis are enriched in a particular Go term. So this is going to be just based on, like, say, PPI plus what we know about a Go annotation. And so we do something similar in terms of ranking, and then we can compare with existing approaches like GSEA and see that Andes actually, again, finds a lot of very clear enrichments there. And so we also have some other kind of downstream applications to highlight that this seems to work quite well. And it works particularly well if it's a gene set that might be under annotated in our existing databases. So the takeaways here are basically computationally, we think actually considering this best match approach once the gene set is large enough, because of course the natural kind of worry people might have is if we're looking for the best match for a particular gene, it could be very much driven by outliers. But our kind of argument and what we find practically in both simulations and in real data is once you have a gene set that's large enough, say we often look at gene sets that are size 10 and above, one outlier can maybe drag like 1 score down or, or make one best match very high, but it's unlikely to be able to do it across everything unless you have a coherent picture across several genes. But this ends up being more powerful than just summarizing very early on, like the results based on a particular centroid. And we can actually apply this idea to set-based or rank-based gene set enrichment, considering the underlying embedding spaces. And very excitingly, this can enable us to bootstrap basically more information about the genes, even if they have no existing annotations. And so we're actually currently trying to develop this into a tool that's more broadly applicable that we think could be very helpful to supplement existing analyses for gene set enrichment, especially in cases where there actually might be more novelty where we don't expect a lot of things to be annotated in existing databases. So just to wrap up, these are the kind of 3 examples of kind of interesting computational projects that we're hoping to, we've been working on. Most of them have this flair of thinking about how we can cleverly integrate 2 otherwise separate modalities or or different databases together in clever ways to derive new insights. So that includes either combining GWAS with single-cell RNA-seq, or combining, say, RNA sequencing splicing information with protein interaction databases, or in this last case, again, thinking about how we can incorporate different representations of protein interactions into basically any type of analysis where we consider gene sets. And then so these are kind of the folks in my lab, and of course our collaborator JP that's helped inspire a lot of the work that we're doing. So thank you all for listening.
Any question?
Yes.
Have you been able to benchmark these tools using different human transcriptome data sets of varying qualities? As we mentioned, we talked mouse data, mouse sequencing is really high quality, human data is very variable. how often does the quality of the human data set affect the performance of these tools?
Yeah, so we've definitely, so I, of course, am highlighting the results that we have using mouse data, and I should really include some of the results. We've obviously done the same analysis. So when there's a tabula miris, there's a tabula sapiens, but the data set definitely is much lower quality. We actually do find a lot of results that are matching. One of the big reasons why I didn't showcase tabula sapiens here is that some of these large human collection efforts, for example, tabula sapiens, actually exclude the brain altogether because it's just like, I think, probably cost and kind of the variability there. There are now more and more brain single cell atlases that we're trying to take advantage of and analyze, but like I said, it's incredibly noisy, but we can still find some of the similar signal. So I think it's always a balance. We typically try to find kind of the comparable human data set to do a lot of the same analysis, but you find that basically combining the two is the most effective, right? So we can have the kind of higher resolution, kind of what we're sure is more clean data, while then looking at the human data to be like, okay, these are corroborated. So sometimes what this ends up looking like is the human data has signal, but it might be weaker. So if we see the mouse signal as, say, very strong, even after multiple hypothesis test correction, we can actually see maybe in the human data there is some weak signal that just maybe didn't achieve statistical significance as per we correct for the more, like the vastly even kind of larger scale number of cell types. And so we're actually also trying to develop further methods that can refine or improve the kind of the power when applied to heavily noisy human data. But sometimes that's like a hint that we can think of, right? It's like, if we're doing kind of completely data-driven exploratory analysis in human, sometimes it's like, okay, it's just really noisy. But if we already have some hints of very strong statistical significance in mouse, maybe we can say like, okay, at least the human corroborates it, even though you wouldn't necessarily independently find the same thing if you just only used human data. So, yes?
So you apply this, your new Go analysis approach to like the brain to get more, how does it impact on what you get out of it in terms of being able to apply and you see the functional applications?
So we haven't actually as much applied it to kind of specific brain studies. This has been kind of this gene set enrichment approach. We're actually just trying to systematically kind of do this across like kind of curated data sets. I think we have yet to kind of apply to more kind of specific studies, and that's like one of our big next steps. Think about like looking at specific studies where maybe there was weak signal and then seeing what types of signals it seems to boost more or another. The other interesting thing is that this last Andy's approach is actually agnostic to the type of embedding that's being used. So what this means is that we can actually make under the hood use say PPI to boost the signal, but we could equivalently here. Let me try to pull this up really quickly. So what we actually did do is actually use Andy's plus actually lots of different machine learning embeddings people have been making. So what's on the left here is actually just lots of different embeddings. So you might recognize some. So when we say like kind of NoTaVec or whatever, it's or PPI, that's like using a protein-protein interaction. People have also made kind of machine learning representations using say like ChatGPT, where we basically take ChatGPT and tell it to make an embedding of the NCBI gene description. So we can also use biomedical literature-based embeddings. And the rationale for there is that, of course, it has like a rough summary of all of Go or whatever, but it also can tell you maybe these two genes actually have a lot of similar kind of descriptions, but have never been co-annotated to the same thing. And we actually found kind of, one of the findings we find is that these text-based embeddings are perform incredibly well, which from a computational perspective, we have to think of clever ways to evaluate this because, of course, there's a lot of potential data leakage because, especially for the ChatGPT-based ones, it's like we don't even know which text it's read or probably all the text. So it's very hard to kind of find like a data set that we're like, oh, it probably never saw this one, so we can actually evaluate it properly. But even then, some of the other, these bioconcept embeddings that I'm showing here, they're also text-based, and those ones have not been continuously updated. So we can take like a time-based kind of holdout. And what we can see is like, and then there's like, for example, you probably AlphaFold, or these ESM, these structure-based embeddings. We can also use those instead as the underlying kind of driver for this gene set enrichment analysis. So we can, I think it's actually a very interesting kind of idea that if we take some kind of complex data set, it'd be also very interesting to swap out the different embeddings and see which things are kind of prioritized or changed more or less, which kind of might give us some deeper insight into, what might be mediating kind of that type of relationship. Yes?
Yeah, I'm just wondering if the seismic computational tool is a free computational tool, so we need to buy license to use
Oh, no, it's okay. no, it's all completely open source. So we actually already have the seismic tools in our package that we've released. It's on GitHub, and just and we're hoping to actually add it to either Bioconductor or CRAN so that people can install it more quickly. But I think it's already live, so it's a free tool to use. So for any kind of
And I have one quick question. I thought the split P was so cool. Did the genes that came up as very rewired, are they also differentially expressed between cancer and
So not all of them. So
or?
some of them are. So I think that's another kind of analysis we think could be very interesting, right? To be like, which ones have strong differential expression, but which ones are like kind of splicing specific.
Thank you.
Thank you.
So the brain tools is
Yeah.
and the